my name is Nathan Davis, and welcome back to my Complex Made Simple series, The Breakdown of Biology. Way back in 1876, a German biologist named Oscar Hertwig came up with a phenomenon called meiosis while observing sea urchin eggs. Now, I bet you're wondering, that word sounds vaguely familiar, and you are not wrong. Earlier, we learned about a cellular process called mitosis. Just to refresh your memory, mitosis is the process of cell division in which your body is creating new somatic, or body cells. On the contrary, meiosis is the process of cell division in which your body creates new gametes, or reproductive cells. Meiosis is necessary to ensure that each offspring will have the same exact chromosomes, which is 46, as their parent, which means each parent will give 23 chromosomes to the offspring. How about I draw this one out for you? This first cell is one with 46 chromosomes, belonging to one parent, which I have 2 here for simplicity. This cell is called a diploid cell because it contains 2 of each type of chromosome, or homologous chromosomes. After the first half of meiosis, we end up with 2 diploid cells, but these are genetically different. Finally, at the end of meiosis, we have 4 daughter cells. These cells are called haploid cells because they only have 1 type of each chromosome, meaning there are 23 chromosomes in each cell. What's great about meiosis is that if you know mitosis, you've already got about 90% of meiosis down. Think about this. Do you remember terms like interphase, prophase, metaphase, anaphase, telophase, and cytokinesis? If so, we're about to look at a very similar process that was displayed in the previous model. The first step of meiosis is interphase. Just like mitosis, the cell is growing in G1, synthesizing DNA in S phase, and replicating organelles in G2. This step is also the longest because it is essentially the cell's ordinary function. Next is prophase 1, in which the nuclear membrane dissolves, spindle fibers form, and chromosomes condense to form tetrads, which are structures of aligned homologous chromosomes. Also in prophase 1, the tetrads will cross over, but I'll talk about that later. Following prophase 1 is metaphase 1, and the tetrads line up in the middle of the cell in a random order. Next comes anaphase 1, when homologous chromosomes are pulled to opposite poles of the cell by the spindle fibers. Independent assortment also occurs, but I'll cover that later as well. Finally, telophase 1 occurs, which is when the nuclear membrane reforms and the spindle fibers dissolve. Each side of the pre-divided cell has 46 chromosomes. Cytokinesis occurs shortly after, and the cell divides into 2 genetically diverse diploid daughter cells. After the first half of meiosis, or meiosis 1, there's actually a resting phase called interkinesis. In more advanced biology, you may learn that there are some certain processes that actually occur. But for now, we're going to say that nothing really significant happens. So let's get back to our phases to see what happens in the following half of meiosis. The first phase of the second half of meiosis is prophase 2. Just like prophase 1, the spindle fibers are going to form, the nuclear membrane is going to dissolve, chromosomes are going to condense, and centrioles are going to move to opposite poles of the cells. Following prophase 2 is metaphase 2, the phase in which chromosomes line up along the equator, or middle of the cell. In anaphase 2, the chromosome's sister chromatids are separated equally to opposite poles of the cells by the spindle fibers attaching to the centromeres of the chromosomes. Finally, in telophase 2, DNA unwinds, spindle fibers dissolve, nuclear membranes reform, and the 2 cells begin cytokinesis. This cytokinesis results in 4 unique haploid cells. Each cell has 23 chromosomes. However, the cells are not gametes quite yet. Our 4 haploid cells need to become gametes in order for fertilization to occur. This process is called gametogenesis. In males, spermatogenesis occurs, and it is when 4 sperm gametes are created, ensuring that 1 sperm will be able to fertilize the egg. On the contrary, only 1 egg out of the 4 is viable in oogenesis. This means that it is a quality process. Now, if you have a sibling, you may be wondering why you may share the same eye, hair, or skin color, but you don't look exactly alike. For example, my brother and I each have brown hair and brown eyes, but we're not identical. This is because of a process called recombination. Recombination includes the processes of independent alignment and independent assortment and crossing over, which all consequently result in genetic diversity. As I mentioned earlier, crossing over occurs during prophase 1 when tetrads form. When the tetrads form, the homologous chromosomes tangle, resulting in some genetic information being swapped. I'm going to use these two sets of cards as an example. Each set represents a half of the homologous chromosomes in a tetrad. Each set contains the same numbers on each card, but they are different colors. When crossing over occurs, the two numbers may switch bags, meaning each bag now has the same number card being replaced, but with a different color on each swapped card. Independent alignment and assortment occur in metaphase and anaphase 1. This is simply the notion that there are many different possibilities of how tetrads can arrange and be assorted. For instance, let's say I have colored cards with case-sensitive letters on them. As you can see, I have two possibilities already based on independent alignment. Once the first half of meiosis occurs, I now have 4 variations. And finally, after the second half of meiosis, I have 8 haploid cells, but each pair is genetically diverse. The final topic I'm going to talk about today is an instance called non-disjunction. Non-disjunction is when a chromosome fails to separate during anaphase, which results in the zygote, which is the fertilized egg from the sperm, having an additional or missing chromosome. This can either result in the death of a zygote or the offspring having a genetic disorder. Here I have a cell in metaphase. When anaphase occurs, one tetrad does not divide, which eventually results in, after meiosis 2, 2 gametes with an extra chromosome, which is called trisomy, and 2 gametes missing a chromosome, which is called monosomy. As I mentioned earlier, if the zygote does not die, or a stillbirth does not occur, a genetic disorder will occur due to non-disjunction. For instance, if there is a trisomy in the 18th chromosome, it will result in a genetic disorder called Edwards syndrome. Edwards syndrome will result in a slow growth rate or a low birth weight. On the contrary, there's only one monosomy that a living human can have, and that is called monosomy X. Monosomy X is a monosomy in the 23rd or sex chromosome of a female. Monosomy X will most likely result in the inability to develop ovaries, heart difficulties, or growth defects. That was a lot of information that we covered in today's video. But just as a recap, today we covered the difference between mitosis and meiosis, what diploid and haploid cells are, the phases of meiosis, gametogenesis, recombination, and non-disjunction. Now you really know what makes you, you. Thank you, and see you next time.